This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on June 1st, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. My guest today is formerly the lab head and head of lab of methods development and associate director of research in the Office of Vaccines and Research and Review at the FDA, and also adjunct professor, University of Maryland and George Washington University, Kostya Chumakov. Welcome to TWiV.
Yeah, thank you, Vincent.
Lot of titles.
Well, you accumulate over your life quite a few.
You were last on TWiV 604. I don't know if you remember. At the beginning of the pandemic,
right.
You were trying to get OPV tried as a temporary way of preventing COVID, right?
Yes.
You talked about that. You never got a trial done, though, right?
Not really. I mean, it raised so many objections, and all the polio eradication people were up in arms and just said, no. Yeah, so it was a kind of quite, it was upsetting, but I can understand,
So you just retired from the FDA, and last week we had a symposium in your honor, which was very nice. Yeah. And I wanted to talk about your life. Not that it's over or anything, but it's a good time to review it. So let's start all the way in the beginning, because you were born in the USSR, right? What year was that, roughly?
Yeah, well, it was 1952, and I was born in a family of two virologists. My father was Mikhail Chumakov, and my mother was Marina Voroshilova. They were both virologists, neurovirologists, mostly studying neuroinfections. Actually, my father became famous when he was a co-discoverer of tick-borne encephalitis in 1937. There was Lev Zilber, who was the kind of more senior Soviet virologist who headed this expedition. So my father participated in it, and he actually got infected with tick-borne encephalitis in this expedition. And he got sick, but he recovered. But he lost movement in his right arm and lost hearing in one of his ears, and 5% hearing remained in the other. So he was handicapped for the rest of his life. And it happened when he was 27 years old. So I mean, it was quite a life-changing event. So I was born into the family of virologists. And
two virologists.
Yes.
Who did they train with?
Well, my father trained with Lev Zilber.
Okay.
He graduated from the medical school, and he was interested in microbiology, so he went to work for Lev Zilber.
Where was that?
It was in Moscow. It was there was an institute. Actually, it's close to Moscow. It was sort of a military microbiological institute, where actually my grandmother, also my mom's mom, also worked there, so they knew each other.
Okay.
So he knew my mother since she was little, and then she also graduated from medical school, and after the World War II, there was a big outbreak of polio in Germany, in the East Russian-occupied, Soviet-occupied part of Germany. So my father was sent there, and then my mom also was in that expedition. She was younger, of course, and then where their kind of romance started, and that's how.
Oh, I see.
So she was his graduate student, and then they started a family, and I was a third of 4 kids. But of course it was probably a destiny to become a virologist, even though I resisted all I could.
Really? You did?
I never wanted to follow in the footsteps of anybody. I mean, it's just, like teenagers are.
Yeah, sure.
Because my father always used to say, oh, the only profession you should choose is medical profession, go to a medical school and so on. And well, I said, no, never ever. So now in retrospect, I think he was right. Because in our field of study, I think having medical education is by far more appropriate than any other education. Because I mean, we're dealing with the human health, viruses. I mean, we're not dealing with plant viruses or marine viruses. We're dealing with human viruses. So you need to understand the physiology and all of that.
Can I ask you about the history of Soviet virology? I was told years ago that the father of Soviet virology was Viktor Zhdanov. Is that correct?
No, it's not. I mean, he was a prominent virologist, but the father of virology, not Soviet virology, was Dmitry Ivanovsky, who was the first to discover TMV, tobacco mosaic virus, because I think, well, at least that's what is written in textbooks, that he was the first to filter this and infect plants. So he came up with this concept of virus.
Okay.
So, I mean, that's how it happened.
Where was he working?
Oh, I wish I remember.
Was he in Russia, do you think? Or
oh, no, he was in it was Russian Empire. It was the end of 19th century.
Right.
So it was kind of way back.
Was Zhdanov a contemporary of your father?
Oh, yeah, he was. I think he was a bit younger, maybe a couple or 3 years younger. Okay. So they were friends, and Zhdanov was quite an interesting guy. He became deputy minister of health at the age of in his early 30s, so which was remarkable. I don't know how he made this career. He was a bright guy, and then after being basically a high-level bureaucrat, he all of a sudden decided that he wants to be a scientist. So he quit the job and became the director of the Ivanovsky Institute of Virology, institute that my father used to be director of at the end of 1940s. And he actually done experiments himself. I've seen photographs of him carrying ultracentrifuge routers, and everybody was wearing white coats, and he wore black, like a blue-collar guy. So no, he was quite an interesting scientist, very interested in virology, and he was one of the people who spearheaded the smallpox eradication. Okay. Along with D.A. Henderson.
Okay.
So he was one of, I think he may even suggested that this is possible. And so he was one of the, brain powers behind this whole effort.
Okay. So both your parents had medical MDs?
Yes, they were both MDs.
So I have in front of us a picture that I think Vadim Agol showed last week.
Yes, right.
Moscow around 1960.
Yeah.
Why don't you describe what's in this picture?
Well, I mean, the thing is that after my parents, both of them, traveled to the United States at the end of 1955, when the Institute of Poliomyelitis was established.
That was in Moscow.
It was in Moscow, yeah. It was, it's Moscow suburbs.
Okay.
So they were both sent to the United States for a few months, along with Anatoly Smorodintsov, another prominent Russian virologist from St. Petersburg. At the time it was Leningrad. And he worked with polio even before my father started doing it. So he was really a polio virologist. And there was 3 of them plus 1 KGB officer who was accompanying them. So I have this all stories from Alexei Sholikov, who actually was assigned by the State Department to accompany them in their travel. So how they became friends. Alexei Sholikov was of Russian extraction, but he never lived in Russia. He was born in China to the Russian expat who fled Soviet Union during the revolution. But he became a prominent virologist himself. Worked in the Panama Canal Zone, and then he worked for a few other institutions in the United States. So he was assigned to accompanying them in their travel of the United States. So they visited all labs that were doing polio research. And mind you, it was early 1956, so it was the height of the Cold War. So it was quite an unusual circumstance. So that's where they met Jonas Salk and Albert Sabin and everybody else who is who in poliovirology. And they became friends. And when they came back, they started this institute and organized production of IPV first. And it quite quickly, I mean, it took them maybe a year and a half to start producing IPV. But then he was actually impressed by the results of Albert Sabin. And
your father was impressed.
Yes. And so my father decided that he wants to start making oral polio vaccine. So they basically interacted with Sabin, and Sabin started visiting Soviet Union. And then I think maybe in 1957, 58, they started organizing a symposium, polio symposia in Moscow. So everybody would come. It was an amazing thing. So all scientists from the United States and from Europe, anybody who was interested in polio would be coming to Moscow because I mean, for some period of time, Moscow became the focus of polio vaccine product because it was the first and the only at the time maker of OPV.
Right, because the U.S. was not interested.
Right. I mean, there was quite a lot of resistance from my former agency, and I mean, for a good reason, because I mean, as everybody knows, perhaps this rollout of the first inactivated vaccine was not without glitch.
Right.
This infamous Carter incident. So there was enough trouble with IPV. So, but then giving live virus to kids, I mean, it was a kind of foreign concept at the time. And of course, there were a lot of questions about how stable it is. And even back then, it was known that attenuated strains are not very stable. They can change. And Sabin knew that they change. But he developed a strategy of how to use this vaccine despite the genetic instability. So that's why having my father on his side, you know, as a collaborator, was a very, I think it was a kind of a collaboration that was really made it happen. The only thing that could make it happen.
So your father invited Sabin to come and help with the OPV production, right?
Yes. Well, he basically said, look, I mean, you have your strains, let's try it.
Right.
So the first thing that Sabin brought this vaccine in his suitcase, you know, just a few bottles of vaccine. So they formulated, I think, in Moscow, and then they had a small clinical trial, and the results were excellent. And so then after that, my father started, you know, developing the production scheme, you know, all these things that you need for mass production, and they very quickly rolled out the first batches of vaccine and started large-scale clinical trials that were also triumphant. You know, it was just basically, it was mostly done in the Baltic republics. You know, now it's independent states, Estonia, Latvia, and Lithuania. I think they started with Estonia, and their polio was almost gone instantly. So it was really a huge success. So that's when they started immunizing the entire country. And actually, Soviet Union was the first country that demonstrated this vaccine can prevent polio. So they immediately started exporting this vaccine into, I think, at the height of this, you know, whole polio campaign, more than 100 countries. So they start, of course, all these, you know, satellite countries such as Eastern European countries and Cuba. Cuba was the first country to eradicate polio in 1962, because I mean,
using OPV.
OPV, because I mean it's a totalitarian country, so they could immunize everybody if you want or you don't want. Nobody cared, so they immunized. So and until now they do it in campaigns, exactly as Sabin wanted them to do. Because Sabin realized that the vaccine is unstable. The only way to prevent cases of what we now call CVD-PV is to immunize the entire population at once, so that everybody gets this immunity at the same time. So the virus that is shed, the genetically modified, you know, virus that regained virulence, would not really transmit to those who didn't get the vaccine.
Right.
So that,
but what about the recipients? Wasn't there any VAPN recipients?
Possibly, but it's such a rare occurrence, so it's not really easy to even to figure out. I mean, it's not until the molecular methods became available, it was possible even to establish that there was a vaccine-associated polio. And Sabin, until his last days, he never accepted this concept. I probably deep in his heart he realized it, but he never openly admitted that this is possible. In fact, the only way to prove that this paralytic case was caused by poliomyelitis was to isolate the virus, and at the time it was not that easy, and then to prove that this virus is vaccine derived. So until there were molecular methods to do it, it was not possible.
So basically, when you're doing mass campaigns, you can't distinguish vaccine paralysis from naturally occurring paralysis.
Well, look, I mean, even because polio, technically, it's acute flaccid paralysis, right? So this is a condition that is not only caused by polio and not only by viruses, but many other, it's basically a symptom. So to prove that this particular case of acute flaccid paralysis is caused by polio and much less polio-derived virus, you need to sequence it and to prove that this is really derived genetically from the vaccine virus. So it's only started happening in the 1980s when it was become clear.
So this photo, you were about 8 years old, right? 1960?
Well, it's probably 8 or 9, perhaps.
That's you in the photo, right?
Yes, that's me.
Then your parents?
My parents and
Albert
Sabin. Yeah, it was taken at the River Cruise terminal in Moscow because at one of these conferences, and it's always was accompanied by some social events. So we went on a short, day river cruise from Khimki. This is a place in north part of Moscow, and of course my parents took me with them. And
so at this time, they must have been doing the trials of OPV already, right?
Already were producing it, and I think they started mass campaigns. So it was those years.
Do you remember any of the interaction between Sabin and your parents?
Absolutely. Yeah, I remember, maybe not at this age, but I remember that trip. And actually, this picture was given to me by Albert himself. Well, already when I lived in Washington, so we met quite often. And he gave me this. He pulled it out from his archive, and he even signed the back of this photograph. So very nice word. To Kostya Chumakov, in memory of our long friendship, for the great scientist, he became such flowery words.
So Sabin tells the story that he went to your father and asked him to do the trials, and then went back with the data to the US and said, "Here, it's a beautiful vaccine," and then they had to use it. Is that fair, you think?
Well, I mean, it's not that simple, because actually there is a very good historical account of all this. Saul Benison, a science historian, he wrote a very nice review. Quite a long, it's not a book, but it's a chapter in some book, very detailed, with the excerpts from their correspondence, letters that Sabin, and all the kind of politics around it. Very interesting. And
do you have a copy of this?
Sure, absolutely. I can send you.
Would love it. Yeah.
And also Albert Sabin himself, he wrote an article saying the history of my collaboration with Soviet scientists. So you may have read it.
That one I've read. Yeah.
Yeah. And that actually was, in that paper there is no controversy, but actually there was a controversy about whose vaccine this was. Because some people in the Soviet Union, and unfortunately including Anatoly Smorodintsov, they claimed that this is a Russian Soviet vaccine. So, and there was a newspaper article saying that Soviet scientists, a great success, and so on and so forth. And both my father and Sabin were quite upset. And Sabin once went to a meeting in WHO where the deputy minister was present, Soviet minister was present, and he raised, and he was quite, direct. I mean, he never minced words. And he said, "I want to ask you point blank, do you claim that this is your vaccine? And so on. And this Minister of Health had the courage to stand up and say, no, Dr. Sabin, I apologize on behalf of the Soviet government that this is misinformation. So they forced basically to retract these stories. And actually, I remember that on the packages, on the cartons with the vials that my father produced at his institute, it was said from Sabin strains. It was clearly marked that it was, because that story was really kind of, it could have ruined their friendship. But then I think in the mid-1980s, my father, he was already kind of, his health was failing, but he managed to submit a nomination to the Soviet government to give Sabin the Order of the Friendship of Nations or something. That was kind of order that they would give to some figures that promoted international, collaboration and so on. So, and then Sabin wrote this. I think it was his acceptance speech. Basically, he made into this article when he described in his own words how this whole thing unraveled.
So let's now move to your career. So you're in this photo. You're very young, but you probably don't want to be a virologist yet, right?
Yeah, no.
When did you change your mind?
Well, look, I mean, I never wanted to be even a biologist. Because it's not. I never liked, biology, because I thought it was kind of, catching butterflies and collecting mouse poop in the forest. All this stuff. It was never appealed to me. I was more like of a kind of. I actually, when I was maybe at the age between 10 and 14, I probably wanted to be a mathematician. So I liked math, and I think I did quite well. But my two older brothers, they wanted to be biologists. My older brother was just, absolutely. He raised some plants, and he knew all the animals and everything. So he was and I, of course, as teenagers, they are contrarians. Yeah. So I didn't want to have anything to do. And my second brother, who was much 1 year older than me, he also wanted to be a biologist. So I didn't. But then what happened, actually, I read a book written by an American scientist, and I forgot the name. That was called something from molecules to cells or something, in which, and I was probably like 14 or 15 years old, and he described, first of all, the structure of atom, hydrogen atom, all the, electrons and nuclei, whatever, and then theory of chemical bond, and then inorganic molecules, and then it going to organic molecules, and then DNA. I said, wow, biology is a science. It's based on, and I started to be a little more interested. So maybe I wanted to be a molecular biologist, never a botanist or, studying real life. And then what happened when I was 14, about to turn 15, in the Soviet Union, in high school. Actually, I call it high school so that the American audience can understand what I'm talking about, because in the Soviet Union it was just one school. From the first grade to the 10th grade, kids studied in the same building, almost the same teachers, changing, of course, year to year, but still it was one continuous process. So and it was 10 grades. In the 9th and 10th grades, I would call it junior and senior, we were supposed to acquire some trade profession, there was some trade education. So our school was assigned to our boys were supposed to learn auto mechanic trade, and girls, I forgot what, something like tailors or maybe cooks, or I forgot what girls were doing because it was separate at the time. So the first we were studying the automobile, how it's done, and then starting in the winter semester, we were supposed to go to the garage of it was a garage of the Soviet Academy of Sciences, and it was winter, Moscow, wind, chilly. I mean, smells gasoline and all this mechanics were running around, muttering and so on. It was just not experienced as a 14-year boy from a good family would want to. And actually, my brother and I, we were in the same grade. And so we complained to our parents, and my mother decided that she will arrange a special kind of exemption for us. So she went to the school director and she talked him into, she said, look, I mean, at our institute, we have a school of technicians, virology technicians. So I promise that if you let them attend this school, we will give them the diploma of virology technician. And that's what happened. So that's how I got into I remember the day, it was March 1st, 1967. So both my brother and I, we were brought to the institute and first started in the histology lab. So we learned how to make sections of tissues, and it was, of course, monkey brains, and then stain them hematoxylin, eosine, and then look at the microscope and so on, and evaluate lesions. So I mean, it was amazing for a 14-year-old boy. Then we went to the laboratory of immunology. We learned how to grow virus, how to do precipitation, and so on and so forth. And then we got to, I think it may have been, it was kind of a rotation. And so when we got into Vadim Agol's lab, which was perhaps maybe April or May of that year, and that's how our collaborations with Vadim Agol started, 1967, so which is like, what is it, 60, 70 something, no, 50 something years, almost. And
your parents must have known Vadim Agol.
Oh yeah, of course, because he worked in their institute. At the time, he was in his late 30s, and he was the head of the biochemistry lab, but he stubbornly refused to study viruses. I mean, first he studied whatever his scientific interest was in, I think, oxidative phosphorylation, some kind of hardcore biochemistry. But then, at some point, he decided to begin working with viruses, and that's the time when I joined his lab. So we were doing sucrose gradients and so on. So I mean, we learned all this technique. And after Vadim Agol's lab, we went to electron microscopy lab. So learned how to work on this Japanese electron microscope, because institute was quite well equipped, because it was a time when polio was a big problem. So Soviet government, I mean, never saved on this. So it was well equipped. So that's how I learned, and I loved viruses. I realized that this is exactly what I mean, because it's a very simple thing. And it's so easy, because I'm afraid of complex systems. I mean, I'm only comfortable in something that I really understand.
So then, eventually, you got a PhD with Vadim Agol, correct?
Yeah. It was later, because I mean, when I graduated from the school, I just turned 16 years old, and of course I got into Moscow University, and first year we were studying some general biological things, but then starting from the second grade, whatever you would call sophomore,
right?
Yeah. So we had to split to different departments. So I, of course, chose Department of Virology because that's where Vadim was. He was one of the co-organizers of this department I think in 1963, when Moscow University decided that they need this new Department of Virology. But I started working with him even in the first grade. I would after the all the classes were over, I would travel to the institute first, and then he actually assigned me to one of his associates in the just newly built building in Moscow University. That was called molecular building. So it was an interesting building, because there were people from chemical department, physical department, biology department, and mathematicians. So it was a kind of multidiscipline institute. It was not called institute at the time, but it was called a lab, but it was quite a big 6-story building. where it was basically all experts from different walks of science. They could collaborate. So it was a very, very one of the best in the Soviet Union, the places to work in molecular biology. So it was just built, and it was not half empty, but there was a lot of free space. So he gave me a room on the 6th floor with a magnificent view of Moscow downtown. It's just, you could, first of all, it's called, at the time it was called Lenin Hills. It was a high bank of Moscow River, and the view was just magnificent. So I would, at night, I would turn off the lights and just look at this beautiful skyline. But I started working, and my first project was I worked with the encephalomyocarditis virus, mouse virus. And my first assignment was to develop guanidine-resistant or guanidine-sensitive or dependent strains, which of course failed because it only works with poliovirus. For EMC, it never worked. But this was my first science project.
You're a college student still, right?
Yeah, I'm a freshman.
Freshman.
Yeah,
Wow.
Yeah, I'm 16 years old, and I'm running my own project. So it was. And working with Vadim was very interesting because, I would call it a negative selection. So, because, he rarely gave specific ideas. He would give, I think you should study this. And then you're on your own. You have to go and read literature, and you have to come up with some experiments. You have to then report what you want to do. He would criticize. He would always criticize. Oh, no, it's not going to work. Okay, yes, it will work. So, and I know that for a lot of people it didn't work because some people were intimidated. So some people didn't have enough, I guess, energy or creativity to. So basically, people who actually succeeded in his lab were really, really creative because anybody somebody who needs some guidance in daily supervision, would never survive in his lab. Yeah. So that's, I guess, the success of his, school. And you know that there are how many people coming from his lab actually became really excellent scientists. So that's a kind of lesson maybe for younger professors how you teach your. I don't know if this can be adapted to the American education system, but that's how it was with me and Vadim. So it was a lot of.
Throughout your whole college time, you worked in this lab under his guidance, right? And then you worked on various coronaviruses, right? EMCV.
Yeah. Well, it was exclusively at the time. It was exclusively EMCV.
Yeah.
I think the main reason why Vadim chose this model, maybe also as a contrarian. If I'm in the Institute of Polio, I should study something else, maybe. But primarily because it was so easy to work with, much easier. Just like Ann Palmenberg told at the symposium last week, it was really, really easy because to grow enough poliovirus for biochemical studies, you need a huge amount, volumes of cell culture. It was at the time it was only monolayer culture, so you need dozens of huge flasks and so on. So it's a very labor intensive. So EMC, we grew EMC in the ascites culture. It was called Krebs2. It was propagated on mice. So you would inject 0.5 ml of this ascites fluid from a previous mouse, and then next week you have a mouse with a huge, belly full of up to 10 mils of, and the concentration was, I think, 3 to the 10 to the 8 cells per mil. So it was enormous amount. So then you spin it down and so on. You can get, dozens or maybe hundreds of grams of cells. So, and the virus could be grown in cultures, as I recall, 2 to the 10 to the 7 per mil suspension cultures. So you could grow tons of virus and then do whatever, milligram quantities of RNA, or if you study virus itself, it was so easy to work with. So that's why we were studying it. And I think my graduate project was my undergraduate, because actually, well, it's hard to tell, because in the Soviet Union, university education was 5 years. So it was somewhere between master and bachelor and master. I mean, Moscow University, at least when I came to the United States, it was accepted as a master degree. In some other university, maybe not that prominent, sometimes they interpret it as a bachelor, but nevertheless, so we had to do experimental project and write a sort of a thesis on this. So my project was to study the reason why double-stranded RNA is infectious. And that was a mystery. Because double-stranded RNA is close to how can it initiate infection? So it was a big problem. So the idea was that we will see what strand of this double-stranded molecule is important. So my task was to separate the strands, inactivate them separately with UV light, and then reanneal and reconstitute into double-stranded and see which one will work. And it turned out that it's not easy to separate the strands. So we somehow managed by using dimethyl sulfoxide and some other tricks. And the result was, of course, that the positive strand was and that was my, undergraduate thesis. But then, since we had such a hard time separating strands, we couldn't understand why. And then, I think it was 1974 or 75, Peter Fellner, who I think is mostly famous for this ribosomal RNA sequencing, even before the sequencing, it was done by some kind of technique with the oligonucleotide maps and so on. He generated a sequence, I think that was 30% error rate, something like this. But then all of a sudden, in Nature, we found a paper he just published in Nature that AMC is a funny virus, that instead of poly-A at the 3' end, as all normal guys have, this virus has poly-C. And since I was trying to separate strains using oligo-DT immobilized on cellulose, so I knew that there is a poly-A. I mean, there was no question that there was a poly-A for me, because we separated plus strands were fished out on this oligo-DT cellulose. So I remember it was Friday, and Vadim came before he went on weekend. He said, well, what are your plans? What are you going to do? I said, well, I came up with a scheme how to localize this poly-C, because I'm sure that it's not at the 3' end. He said, oh, Kostya, don't even bother, because they already figured it out. So I'm sure that his second paper is already in press and so on and so forth. And I said, well, no, I will still do it. So I basically I crushed the RNA molecule by boiling it for like 10 minutes. Yeah, so it was fragmented into small pieces, random length, and then I separated those that contained poly-A, absorbing on the oligo-DT cellulose, and then whatever I eluted from the cellulose, I absorbed on oligo-DI that would bind poly-C. So, and then put it on the sucrose gradient, and to my surprise, all the fragments were distributed across the entire sucrose gradient, but those that passed through the two columns were sharp full-length molecules. So it was just an unambiguous result showing that poly-C is on the 5' end, not 3' end. So it was so shocking. Wow, that was so easy. And just one weekend.
You said you worked all weekend to show him on Monday, right?
Yeah, on Monday he came to the lab and I showed him, and he said, okay, so where do we send this paper?
Great.
So I was very proud.
And you were an undergraduate at this point.
Yes, I was undergraduate. Yeah, and this was my PhD thesis, because I worked for a few more years, because Vadim wouldn't let me actually defend my PhD. He said, no, a little more, just get a little more results. But then I broke my arm. I fell, slipped on the ice and broke my arm, and I couldn't work in a lab. So I said, Vadim, I will use this month that I'm still in the cast. I will start writing my PhD thesis. I said, okay, all right. That's how it happened. But the result was generated in one weekend. And actually this paper was, because we were trying to rush it to be published. It was early 1976. And the Soviet, and of course we wanted to publish it in a decent journal, something that will be fast, but still English language, not in the Soviet, because nobody would read it. We wanted to be first before Peter Fellner would publish his results. So, and it was not easy. In the Soviet Union, there was a system that you were first supposed to publish your findings in Russian. And then, if you want to send it to English language or foreign journal, you will have to pass through the special committee that would review this, and you will have to fill out the form in which you state exactly that this paper contains no new information, and therefore it can be published in the foreign language. It was absurd.
That's funny.
But the post office would not accept the manuscript for shipping. There was no internet. You couldn't upload it in PDF. So there was basically the only way to publish it fast was to smuggle it out of the country. And fortunately, at the time, Carlton Gaidushik was visiting Moscow. It was the end of May 1976. And I told him that we have this problem. I have a manuscript ready for publication. And he said, well, I can help you because I'm going to Paris now and I will meet Jacques Monod. And he is the editor of the Biophysics and Biochemistry Research Communication, BBRC. So he took the paper and it was published. So Jacques Monod submitted it on May 30th. Because I have a letter from him, May 30th, 1976. On May 31st, he passed away. So it was the last paper that he submitted.
Wow.
So it was it was so weird. I only learned that he passed away a few months later and said, let me look at the letter of acceptance. And it was kind of the last day of his life.
That's a good story. So during this time, also I understand Eugene Koonin and Sasha Gorbalenya were in the lab and working with you, is that right?
Well, yes. I think Sasha joined in 1975. At the time, I graduated in 1973. And so he actually came to the Institute of Polio, where I never worked there, I was never an associate of the institute, but formally I worked in the Moscow University, but the best lab was, Vadim's lab was in the institute. And Vadim said, well, I think it's time for you to start your group. And there is a guy who was sent to us from Novosibirsk, and at the time Sasha graduated from Novosibirsk University, and he was sent to work to this newly formed institute that's called Vector. It was an institute that was supposed to be involved in military development, some kind of bioweapons development. So maybe not exclusively, but the large part of it was. But since there was no building yet, so they were only started construction. So all the new associates who just got from college, they were sent to different places in Moscow to basically to learn and for training. So and that's how Sasha and I started working together. And then Eugene Koonin just graduated from our department of virology also, and so he started working also about the same time. So there was three of us working in the same room, and it was kind of it was a very good time.
So you were getting independent by this time?
Oh yeah, no, I was always independent. That's the thing about Vadim's lab. So because he treated all his associates it was not like there was a hierarchy. So I was helping Sasha and Eugene, so they were sort of in my group, but they also interacted with Vadim directly. So it was not like in some labs when you basically a lab chief doesn't really know what the kind of everybody is doing. So it was not a big lab. I think there was maybe 10 or maybe sometime 15 people. So it was not a huge lab. So that's we started working. Initially, it was exclusively a lab work because Sasha's first project that I came up with, because I mean, when I was told that Sasha will be working with you, and Vadim said, well, think about what he will do. And I said, well, I mean, now we know that this poly C is on the 5' end, let's figure out the function. So let's try first see what proteins bind it. So Sasha's project was to use all this oligo-D-C, oligo-D-I, oligo-D-A celluloses to basically to do affinity selection of proteins that bind it. So both in virus-infected cells and normal cells. So that's how he would pass the protein extract through the cellulose and then wash it off and then run gels, one-dimensional, two-dimensional, and so on, and look at this basically spots and would compare and look at the difference. And I was impressed how perceptive he was. He was, look at this. You see this tiny spot here? It's not there. I said, oh, really? Do you think so? but he really was so focused, and he had this ability to see tiny differences. So that's, and we published this, I think in Virology, some of these findings. And then Sasha was sent back to the Vector Institute, where he basically refused to sign the security clearance. Because, I mean, of course they required, because it was a special kind of institute. So he didn't want to have anything to do with this, so he was sent to the basement to open crates with furniture. So, I mean, they couldn't fire him, but since he was not, he didn't get the security clearance, he couldn't work in the lab. So that's, it was the time when you and Eckhart published polyonucleotide sequence. So Sasha, during the lunch break, he would look at the sequences, and he with another guy, Vladimir Blinov, they would basically, without any computer, they would gaze at the sequence and see some things. For instance, they saw something that resembled protease, and then at some point, Sasha was able to come back to Vadim's lab and basically demonstrate that indeed this protein, that this PP22 of EMC is indeed a protease. So it was, I mean, I was shocked, because I mean, probably it was the first example, at least that I know of, that you find something in the sequence and then prove the biological function just by looking at the structures, looking at the motifs. And it was 1977 or something. It was really early. I mean, there were no computers in the Soviet Union. Maybe even the United States, they only started.
Yeah, that's right.
Only mainframes. Yeah.
So last week, Eugene Koonin told the story where he's working. You tried to teach him to do biology experiments, and he couldn't do it. So you had told him to work on a computer, right?
No, I didn't tell him to work on a computer. You know, because look, I mean, not everybody is the same. You know. Eugene is brilliant, absolutely. I mean, and I enjoyed working with him because, I mean, one of his first projects was a cell-free system for RNA replication. So so we worked together on this, because he was still sort of under my wing, so to speak. And we quickly realized that nucleoside diphosphates are incorporated much faster than triphosphates. And first we will say, what's going on here? Because I mean, it's counterintuitive, and even monophosphates were incorporated better than triphosphates, which is counterintuitive because everybody knows that triphosphates are used as a substrate by by any polymerases. But then we came across a similar finding for DNA phages that actually were interpreted that there is all the enzymes, kinases that generate triphosphates from monophosphates, diphosphates, triphosphates. They're probably organized in a certain pipeline, physical pipeline, so that to get to the active center of polymerase, you need to enter as a monophosphate. Because for triphosphates, it's very hard to get into because it's just structure release. So I think it was an interesting observation, so that RNA polymerase complex of epicornoviruses are organized in such an interesting and complex way. So basically, viral polymerase is integrated with cellular kinases. So I mean, it's. I think it's a fact that is not really appreciated, that the internal structure of a cell is not it's not a bag with enzymes that are just
that's right.
Floating around. So they're structured.
Yeah, that's right.
So, so he could do experiments, but then when the sort of nucleotide sequences started rolling in, and then when Sasha's success with this discovery of protease and the analysis of sequences, he got so much interested in this. So, and at the same time, I also drifted into into sort of bioinformatics, even though there was no such word at the time, because they think the way it happened was also funny, because there was a guy in the Institute of Polio who studied the virus, and he thought it was a virus, and he even said it's probably enterovirus because it's about 30 nanometers in size, capsid, and it's filterable, and it was isolated from a case of Vilusk encephalitis. So it was a mysterious illness in Siberia when people had symptoms similar to Kuru, this slow virus infection that is now believed to be caused by prions. And so, of course, there was an expedition sent to this place, and they isolated quite a few viruses that most of them were completely irrelevant. But there was one virus or agent, they called it KPN, as initials of the victim of this Vilusk encephalitis. So they studied about for 10 years. So they would propagate it in cell cultures, and they couldn't figure out what it is, because under the microscope, they say, well, it's a virus that causes such a dramatic change in cellular morphology. I mean, the cells are unrecognizable. But they couldn't figure out. So at the time, I was running gels like every day, and so this Andrei Karavanov, he asked me, do you want to look at this? Because you are working with enteroviruses, it will be probably easy for you. So the first gel that I ran was weird, because it was clear, I've never seen such picture. There was 3 closely located bands of RNA, about 20S. There was a sharp band on top that looks like DNA. DNA always runs as a sharp band. And down at the bottom, there was a kind of diffuse bands, something looked like tRNA, and there was a sharp band of something looked like 5S RNA. I say, well, it's not a virus. Or if it's a virus, it's a weird one, because it has both RNA and all this collection of things. So I decided to sequence this 5S RNA because it was easy. And I sequenced it. There was a method that was developed by DePrePT. It was similar to Sanger methods, but slightly different chemistry. And when I got this sequence, I had no computer at the time. But there was an issue of nucleic acid research that published every year all known sequences. It was not a very thick issue, so I basically opened the chapter where it's 5S-RNA, and I typed on the strip of paper this nucleotide sequence, and I was basically moving it, and I found the sequence that was 100% match. All 121 nucleotides matched exactly. And it was on the side, it was called Acanthamoeba castellani. It is amoeba. It's a free-living amoeba that occasionally causes neurological disease. So, I mean, it was just amazing. I was so... And then, of course, when I started reading about it, turned out that 28S RNA is split in half. So that's why it has 3 bands of. So it was everything explained beautifully. And then when I took this virus supposedly to electron microscopists, to the Institute of Parasitology, they said, "Where did you get such an excellent sample of amoeba? Can I use it for my lectures?" So it was classical, and the dramatic transformation of the cell morphology was very simple. Because it was amoeba, it was not a cell culture, and even if you, try to use this focus, fine focus, you can see that the amoeba were sitting on top of the culture, so it grew perfectly. So it used cell culture as a substrate, but it was not inside the cell, but on top of the cell. So I was shocked that it was so easy by sequencing to resolve something, a mystery that existed for 10 years. And while I was still kind of unsure what it is before I sequenced, I started collecting samples from all over Moscow, all kind of bacteria and chlamydia or all other organisms. And since it was so easy to sequence, I accumulated a huge database of 5S-RNA. I think at some point I sequenced more 5S-RNA than anybody else combined. So I had a huge database, and of course I didn't know how to analyze them. I started asking around. Does anybody have any software to do it? And first of all, there were no computers. There were mainframes because Soviet Union was under embargo. There was no way you can import anything. At the time, there was not even Macintosh. I think that Steve Jobs was still in high school. I mean, it was a time when there were no personal computers. But there was a guy working in the institute who actually they did have a personal computer. It was called YCAT. It was way before Microsoft or Apple. It was Unix-based computer that was smuggled in again. I mean, by some Swedish salesperson who sold them some equipment, but and there was some change that he owned them. And so he asked, "What can I bring you?" They said, well, "Bring us a computer." So he just brought it in a suitcase. So nobody asked questions. So I had access to this computer, and I met a mathematician who said that he was a topologist, Sergei Yushmanov, and he said, well, he just got his PhD for some mathematical work on graph theory. And I said, I visited his seminar, and he was talking about the phylogenies and so on, and reconstruction. And I actually, after the seminar, I came up to him and said, look, can you write a program? Because I have a computer, you know the algorithms. So he said, sure, let's work together. He was about my age, so we were all in the early 30s, so enthusiastic and so on. And so we started writing. He wrote a computer code that wouldn't work. And he would work from home. You know, mathematicians, they don't want to go to offices very often. So I would call him and say, look, I mean, it doesn't work. It gives me such an error message. Oh, I'm so sorry. I mean, on line 156, there is a comma and there should be a dot, or period. I actually go back and I say, okay, I replace it, and it works. So I started looking at the code, and well, it's almost in English. If, for, and all this. So I started doing it myself. So at the end of the day, I learned how to do this computer programming. And I wrote this quite a substantial suite of programs myself, including reconstruction of phylogenies using different algorithms. And Sergey and we came up with some new algorithms that Eugene mentioned last week. So we published, but only in the Russian literature, because I mean, we didn't have time to write papers and so on. So, but we created this suite of software products and used it quite efficiently, because I mean, then by then Eugene got interested, and we used my programs to reconstruct all these phylogenies, polymerases, and so on. That's how these papers that we published together came about. And then Leonid Brodsky, a mathematician who worked at the same. That's the beauty of this place. I mean, you can find anybody, somebody who had a smuggled-in computer, somebody who is a mathematician, and so on. So once we met, and he said, "Well, I heard you're writing biological software." He said, "Let's maybe try to sell them. Let's start a company." At the time, it was called cooperatives. I mean, it was just Perestroika just started, so they allowed some kind of private enterprises. I said, "Wow, who would need this? I did it for my research only." Yeah. So, but nevertheless, we ended up forming this software company that was called GenB. And we would go to another city, to some biological institute, give a seminar, and then sell it. So that's why the last few years of our life in the Soviet Union was kind of interesting. It was a spin-off, basically.
Yeah, cool.
So that's how this whole bioinformatics thing started with Sasha and with Eugene. But I actually very soon, in a year or two, I moved to the United States and then started doing very different things.
So why did you come to the U.S.? What's that story?
Well, the story was that I told you that, like, when I was studying this microbial phylogeny and classification, and at the time, of course, Carl Wöse was developing this ribosomal RNA-based phylogeny, and most microbiological journals, they stopped accepting papers describing a new species without phylogeny. Yeah. Rightfully so. Yeah. I mean, at the time it was kind of absolutely a requirement. Since the rumors about me doing this spread around Moscow, I was asked by, there was an Institute of Microbiology and the Soviet Academy of Sciences. So the institute was famous for a lot of expeditions they would do to some remote places and finding microbes here and there and everywhere. But they had a problem with publication, so they needed somebody who would do it. So I would first help them to do it, and then the institute director changed. So it used to be a kind of a guy in his maybe late 80s. I mean, completely. I mean, from a totally different era. He wouldn't understand much about molecular biology at all. So he was a kind of, well, anyway. So he basically, they managed to get him out and then appointed a new director who was in his, I think, maybe early 50s, so much more, kind of modernized, westernized, and kind of quite reasonable. So they talked, all the associates, they talked him into inviting me to come to their institute. And at the time, I was doing all different things. Vadim didn't like it at all, because he said, well, you're not doing virology anymore, you're doing all this phylogeny, you're doing computer programs and whatever. You're not interested in virology anymore. So basically, I was still in his department in Moscow University. But sort of our relationships, we basically split ways for a while. He was doing his thing. I was helping him a lot. For instance, I wrote for they worked with OLMQ at the time on Galina Lipskaya and Olin, they worked on this oligonucleotide maps to distinguish vaccine-derived viruses from wild viruses and so on. So they needed, basically they would run these two-dimensional gels with spots telling them the difference, but it was not easy for them to kind of match each spot with the sequence. So for me it was easy. I just wrote a computer code that would display on the screen the theoretically derived oligonucleotide map. So that basically you put in a sequence and you get the map on the computer. So for them it was very, very important to have this kind of software. So that was the only collaboration I had with Vadim at the time. And finally, I got this invitation to become a lab chief in this Institute of Microbiology. It was a huge honor because the previous chief of this lab was an academician, well established, and he was a former director and so on. So for me, it was like 35 years old. It was a huge honor. I mean, it's just to become a lab chief in such a prestigious place. So I accepted this offer, but I quickly realized that it was not what I was bargaining for, because they had no money. It was the end of the Soviet Union. Everything was falling apart. Mikhail Ivanov, who was this new director of the institute, he promised to me good funding so that we could have no problem. But we had not even the furniture. We got a new 3 rooms without the furniture. And he told me, well, you can hire 10 associates. So I invited everybody whom I respected of my former students, including Eugene Kunin, who was my deputy at the time. So we were very enthusiastic. I was the oldest, 35 years old. The rest of the bunch were in their late 20s. So we were so excited that we have such a young lab. I mean, we will, basically move mountains and so on. But then we were sitting in the bare walls. I mean, fortunately, we had some chairs that we could have seminars. And also, fortunately, each of us retained some connections to their old places where we came from, and including myself, I still had the lab in the Moscow University, and it lasted for a year and a half. And I realized that I'm not going to spend my most productive years, basically sitting in these bare walls with no furniture and fighting windmills. I mean, it was just not. So I started thinking about going to the United States. I tried many times before, and I was never let. Eckhart Wimmer invited me twice, a few years back, during the Soviet Union, before Perestroika. I was never allowed to travel. Probably I was not a member of a communist party. I probably told too many jokes about Brezhnev. I mean, I would love to see my KGB file, but it probably was extensive. Okay. So I was never allowed to travel. But then the times have changed, and I was already a lab chief, so on. So it was easier. So I thought maybe I will try. So I wrote a letter to Carlton Gaidushik, who was good friends with my parents, and also we became friends. So I mean, and he said, sure, absolutely, no problem, because I mean, I've seen your paper about this KPN agent being an amoeba. So why don't you bring the samples with you, and we'll continue this research. And then at the same time, I actually was introduced to a lady, Ines Levenbuk, who was also a Russian expat. So she immigrated from the Soviet Union. She used to work at the Institute of Tarasevich Institute, which is a counterpart of SIBR in Russia. So it was a Institute of Control of Biological Products. But she immigrated in 1979, and she started working for SIBR. So it was the first time that she came back to Moscow from the United States, because, of course, they left forever, because there was no way. I mean, if somebody immigrated from the Soviet Union, they would never allow to come back. But during the Gorbachev time, it was relaxed, so she came back. And we were introduced to each other. We spoke for 5 hours, and obviously we liked each other. And she said, well, look, if you have somebody, if you have a kind of a young student who would be interested in coming, because I have an interesting project that I want to start, so if you have a candidate, just give me his or her CV. So I gave her a CV of one of my young associates, and she left. And then I went on vacation. It was August, and when I came back from vacation, I found 2 telegrams. You know, the old-fashioned telegrams with the strips of. Kostya, I found a good position for you, not for your guy, but for you. It's a 2-year position at FDA, so please come and so on. Let's start working on this. And when I went to my institute director, he said, "Oh no, you cannot go for 2 years. You're a lab chief. You must be here. So, but you can go for a couple of months and just, you know, make contacts and just look around and so on." So okay. So a month later, I was in the here, and the first day I walked in the lab, I said, "No, I'm not going back." I'm not stupid, because I mean it was, and the business was Carlton. It dragged for so long, and he was traveling here and there, so it never actually materialized. So that's how I ended up in Sibir.
What year was that?
It was 1979, the very end of 1989. Sorry. So, and then of course the project was on polio, and it was the first time I touched polio. So it was ironic. I tried to escape this fate for all my life, and ended up doing things that my parents did. So that is.
So when you were here now in the Washington area, Albert Sabin was here, right?
Oh yeah.
So you must have been in contact with him, right?
Very often, yeah. He lived on New Mexico Avenue, so I would be a conduit between my father and him. My father would send me letters in Russian. I would translate them in English, and I would go to him and meet his wife Eloisa, and we would spend time. They would invite us for dinner, and we would invite them, and so yeah, no, we interacted. So he was a very nice guy. I mean, personally, he was very. I remember once he invited me to a Cosmos club that you visited recently, right?
Yeah. Last night. Yeah. Was it last night? Yeah. Wait. Today is Thursday, right? Yeah. It was. I gave a talk at the Cosmos Club. Yeah. His picture is on the wall because he was a member.
Absolutely.
Yeah, that's right. Yeah. So let's talk about some aspects of what you did here at the FDA. One of them, I mean, you got involved in working on polio vaccines. It was OPV mainly, right?
At first, yes. Yeah.
And you developed, and this is how I got to meet you, through this test that you developed for looking at the 5' non-coding region changes and making sure that production lots of vaccines had the right. It was called MAPREC, right?
Yes.
Mutant analysis by PCR and restriction enzyme cleavage. So tell us what was the general problem that you were trying to solve?
Well, the problem is that the strains of oral polio vaccine, Sabin strains, they are genetically unstable. So when you grow them, because I mean, look, they are attenuated, meaning their fitness is reduced intentionally. So that they don't cause disease. So they're basically attenuated. But all live organisms, they want to reproduce, so they actually like to increase fitness. So this is a general biological law that all organisms, they want to increase their fitness and live as much progeny as they can. So of course this happened, and Albert Sabin knew this. I mean, because he tested his vaccine. I think that he used like 10,000 monkeys before he actually was sure that this vaccine can be used in children. So he was quite thorough in his experiments, and he knew that the virus, if it's grown under inappropriate conditions, and virus that pass through the gut, that is excreted, has a higher virulence than the original strain. So it was known well before molecular biology. But of course, at the time, nobody knew what was the molecular basis for this de-attenuation. So that's why he required and he demanded that anybody who produces vaccine from his strain must do the monkey neurovirulence test, meaning that each lot of vaccine, which consists of 3 serotypes, right? Each of the serotypes must be tested in monkeys. So the first test was they inoculated intraspinally and intracerebrally at different virus concentrations, and then they would sacrifice monkeys, make sections of the CNS, and they look at the lesions and so on, and compare results for a group of monkeys inoculated with the test vaccine with a group of monkeys inoculated with the reference vaccine. So then, after a collaborative study that was conducted in the early 1980s, they came up with an improved method which streamlined the process, but the principle was the same. You have to inoculate a lot of monkeys with vaccine and the reference strain and compare the results. They developed a special scoring system, grading lesions and inflammation on the scale of 1 to 4, and then averaging it out. I mean, it was a very cumbersome test, but it was, first of all, inhumane, using so many monkeys, because for one lot of trivalent vaccine, you would need 200 monkeys. And in many countries, the test was supposed to be run by a manufacturer and by the regulatory authority. So, I mean, it was extremely expensive. Monkeys were, at the time, I remember, a few $1,000s per monkey, because they were bred in a special colony in the United States. In other countries, they were imported from Africa or from Asia. So it was really, I mean, something that everybody wanted to get rid of. I mean, but without this test, it was not possible to control and make sure that the vaccine is safe, because it is not exactly a safety test, but it's sort of indicative of virus stability. So if a vaccine batch was prepared in inappropriate conditions, the thinking was that it may not be safe, because we know that these mutations are increased virulence in monkeys, and by inference it was decided that maybe it's also connected to virulence in humans.
But we don't actually know, because no one ever released a lot of...
No, actually it's not,
yeah.
It's not true. There were instances when a vaccine lot was released, and then later on, upon retest in another country, would be shown to be inappropriate, unacceptable in terms of monkey neurovirulence. So that actually helped us to develop this MAPRIC.
That lot, did it cause more paralysis than you would expect?
Yes. No, it did not. No, it did not result in any single case of VAPP.
Which maybe tells us the monkey test is irrelevant for humans, right?
Well, it's not irrelevant. It's a consistency test. It's a consistency test. It may have nothing to do with virulence in humans, because every single case of VAPP was caused by a vaccine that passed the test.
So vaccine-associated paralytic polio, right?
Right. I mean, all these unfortunate, very rare unfortunate cases of vaccine-associated paralysis were caused by vaccine that passed the monkey test with flying colors. I mean, that's... it is not a safety test, but it's a consistency test, which is also important. It's very important, because every consistency is it's a cornerstone of good manufacturing practice. I mean, you have to be able to produce something consistently, time after time after time. So, but never, and especially for knowing that this is a consistency and not a safety test, of course, everybody hated monkey test. And by then, Phil Miner and Jeff Almond and their group, they have identified mutation that was primarily responsible for this deattenuation. It was in the 5' UTR. And later, other groups demonstrated that similar mutations in other serotypes of polio, because Phil and Jeff, they worked with type 3. Also are located in the same hairpin structure called domain 5 in the 5' untranslated region, which is, we know now, that is part of iris, internal ribosome entry site, a very important element for virus life cycle, for protein synthesis. Inessa came up with this idea. So maybe this lots that fail a monkey test, maybe there is increased content of revertants. And she actually talked with her son, who also graduated from our department at the time. I think he worked in MIT. No, at the time he moved already. He graduated at MIT from, he worked with Alek Warshawski. And then he got his own lab in the University of Chicago, Illinois in Chicago. And they actually discussed all this because he was a molecular biologist, and he basically, I think that he gave her this idea that maybe this can be done. So that was the project I was invited to work on. And when I came, very quickly we realized that by then they had some experiments that demonstrated that this PCR, actually the way it was done, that one of the primers was located next to the this 472 position, and it was modified at the 3' end so that it would create a restriction site that would be toggled on and off by this mutation.
Right.
So the normal vaccine sequence would be cut by HNF1, and the mutant would be cut by MBO1. So it was very easy. So they have done some preliminary experiments, and when I came, I said, guys, I mean, we're never going to get enough sensitivity unless we use radioactivity. So we started labeling one of the primers with gamma ATP, and then run the gels and look at the X-ray films, and so on. So that's how we basically, and it took less than a year to demonstrate that first the method works, and second, fortunately, we had close to 300 lots of vaccine made by different manufacturers. Mm-hmm. And we knew they were all tested in monkey test. Mm-hmm. And we knew which one are failed and which one are passed. So that's why for us it was easy to test them all and then plot this chart that Phil Miner showed at the symposium last week. And it was remarkable. I mean, realization that all batches that passed, they were had a content of mutants less than 1%. Mm-hmm. And the vaccines that failed, they were above 1%. And there was quite a clear cutoff line that was determined by the US neurovirulence reference, not surprisingly, because this was the reference that defined a pass or fail result. Mm-hmm. We published, and actually Albert Sabin, I called him and I said, well, look, I mean, we have this paper. Would you consider it for publication on PNAS? And he said, no, Kostya, I'm so sorry. I decided I no longer will submit anything because I'm getting old and so on. But please send me the paper. I sent him the manuscript, and a few days later he calls me, Kostya, I read your paper and I decided I will make an exemption for you. but only on condition that you will accept all my edits, all my changes that I will suggest. I said, sure, I'll be honored, and so on. So I spent a few days in his kitchen, in his apartment. He would use this red pen and would cross out every instance of the word mutation and replace it nucleotide change. Okay, all right.
That's not bad. That's actually not a bad thing.
That's probably, in retrospect,
it's probably right.
It's right. Yeah. I mean, I'm not much into semantics, but I think he just wanted to avoid because
that's why we call them SNPs instead of mutations,
exactly.
right? Because it's a polymorphism.
It's a polymorphism. And he said, well, mutation has a negative connotation, and in this case, you don't know, you don't have a phenotype. I mean, even so he was correct. I mean, he was so smart. It's amazing. He understood all this concept of this quasi-species way before anybody, I mean, even thought about this. Not even a term, but in this, that's how he selected his strains. Because, I thought it was a kind of alchemy, basically he passes here and there, different cultures, like purified. I mean, just like most people think it's a kind of empiric process. It was not empiric process. He knew what he was doing. And he explained to me how he came up with this idea. He said that viruses are a mixture of different particles with different genetic potentialities, as he called. So there's always a mixture. And it's a matter of selecting those that pre-exist there. And I realized that when you passage virus at a low multiplicity, they retain their properties. If you select at a high multiplicity, do rapid passages, they quickly lose their he didn't use the word fitness, but he said they lose their virulence. Mm-hmm. So that's why he passaged them 30 rapid passages, meaning that he would infect with quite a significant MOI, multiplicity of infection, and would harvest 24 hours later.
Yeah.
Regardless of CP, and then do the same. And he passaged 30 times, and it worked. So he selected the virus that contained 472U instead of C, something that virus would love to get rid of. And the same goes for this another mutation in VP1 nucleotide 6, 2493. Right. Also, I mean, this is a mutation that is reverts back like in an instant. 2 passages and it's almost completely reverts back. But somehow he selected it. Mm-hmm. So I mean, there was something to his reasoning. So that's why, and it worked for all 3 strains.
So why is, if your molecular test shows that the amount of C correlates with monkey neurovirulence, why isn't it a safety test then? Because it's the amount of C will then predict what happens in people, or we're not even sure about that?
Well, I don't know. I mean, I think, again, this is a matter of consistency. We also made an experiment that also puzzles me. It makes me think that there's something else in play. Because, I mean, for instance, we would inject a monkey spinal cord with a perfectly good vaccine.
With a U at 472.
Yeah, absolutely. Quite unacceptable with low levels of all these mutations, and then sacrifice them a day later, like 24 hours later, and would isolate virus from the spinal cord, or just do PCR on the homogenate. It would completely revert. It means that if you inject into monkeys, into spinal cord virus with 100% 472C, they would be paralyzed instantly. But 24 hours later, all virus that you can recover from the spinal cord is already mutated and they're not paralyzed. So,
and in fact, in kids, in every kid who gets, within 48 hours it's reverted, but only a few get paralyzed, right?
In kids, it's not 24 hours, it's more like a week.
Okay.
But it's a gut. It's slightly different, because poliovirus rarely gets into the CNS at all. But in this case, it was spinal cord. It was where the motor neurons are. And my interpretation now, there was some innate immune response going on. So that virus would revert, and because you cannot imagine that there will be a specific immune response, in 24 hours, too soon. Yeah. But there was something that happened after inoculation that would protect neurons or CNS in general from this virus that was breeding inside the CNS, and nothing would happen. So it's still to be explored, I guess.
Well, unfortunately, I don't think we will be able to, because people can't work on polio very readily anymore.
Oh, that's it. Yeah, that's the end of the road.
It's a big problem, because I think there are a lot of questions that need to be addressed.
Yeah, absolutely. Well, look, Vincent, I think that that was one of the reasons that I decided to move on, because, I mean, it became so difficult. It's just an uphill battle, and this polio eradication is not helping. I mean, it's like I said last week, I mean, they did not eradicate the virus, but they eradicated poliovirologists. Because, I mean, a lot of people just voluntarily say, okay, the hell with it. I mean, we don't want to mess with this stupid containment, which scientifically makes no sense. And it's now all in the hands of bureaucrats. They don't really care about science or even public health, because, I mean, this program, I mean, it's been in the dire straits for many years, but now 35 years later, we're still
about
not
the
eradication program, and it's like nobody wants to think, stand back and think about what are we trying to accomplish. So it's like a Flying Dutchman, you know, the ship is moving and nobody is on the ship. It's moving and people are doing, I mean, a lot of people are involved and nobody's thinking about the strategy. And it's a shame, but here we are.
So I wanted to end on talking a bit about eradication. Well, first of all, before I do that, the MAPREC test you devised, was that ever used did it ever replace the monkey test?
No, unfortunately not. I mean, it did not. We have, like the first year when we developed, we published a paper, and then, of course, WHO got interested, and a lot of companies also were excited about this possibility. And so we started, WHO started sponsoring collaborative studies with the participation of vaccine manufacturers and national control authorities. And it lasted for almost 10 years because it was started with type 3, then type 1 and type 2 and so on. And it was it's quite a lengthy process. So we figured out we basically identified a lot of glitches, you know, technical tricks that you need to be aware of. It's remarkable. It was a very telling experience. How the research grade test differs from the regulatory grade test. I mean, the rigor of evidence, the level of certainty must be much higher. So, but one way or another, at the end of 1990s, it was recommended by the Expert Committee on Biological Standardization as the preferred in vitro test, because our arguments that it predicts almost 100% on the failure in the monkey test fell on basically deaf ears. Because I mean, at the time, this committee was mostly composed of old-time, biological standardization types. They knew almost nothing about molecular biology. And I remember when I was presenting, somebody asked me, are you telling me that by looking at this X-ray film on these black spots, you can tell me that it's safe to inject into my grandchild? I said, well, I think I do. Oh, no. We still need, a monkey test. But fortunately, at the time, in our lab, in S11 book and Eugenia Dragunsky, they started working on using transgenic mice as a replacement for monkeys. And also they went through this rigorous and laborious and time-consuming collaborative study process to basically implement it. So it was also accepted by WHO as a replacement for monkey test in conjunctions with MAPREC. So basically, these two methods that were developed in our lab became the gold standard for OPV lot release.
And so now OPV that's made, and I understand there's just one country in the world that makes OPV, is that?
I'm not sure. No, probably more than one.
Because there's a lab in Indonesia.
Indonesia. I don't know if Pasteur Mirieu, I mean, the Sanofi still makes it.
Okay.
Maybe they do it. GSK, I think they used to make it until recently. Maybe they're not anymore. But I think that they make it also. Indonesia, yes, definitely Indonesia. But I think also somewhere in China.
Okay, but they use MAPREC and transgenic mice,
yes,
no longer the monkey test.
No, I think that some companies still use monkeys because for some of them it's cheaper to use because transgenic mice are not easy to breed. They're produced in Japan by the Central Institute of Experimental Animals. They're quite expensive, so that's why I think that some countries still use monkeys.
Okay, got it.
All right. Oh yeah, Russia still produces OPV. And they use monkeys. So, yes, I mean, it was implemented. It was implemented, and now it's being used. But now we're trying to get rid of it. I mean, MAPRIC.
Yeah.
Because of course it's a 20th century method.
Sure.
And it requires radioactive isotopes. You can use fluorescence, but I think it's still preferable to use radioactive isotopes, which is now difficult for regulatory purposes reasons.
Yeah. Okay.
So I mean, so that's why after we have done this with MAPRIC, and it was one other, actually, I think more legitimate argument than just looking at the black spots, was that you look at just one nucleotide. How can you tell us that there is no reversion at some other site?
Sure.
I mean, that was a legitimate argument. That's why we started exploring different methods, mass spectrometry and microchip hybridization, all these new things that actually came along the way in the early 2000s. Mm-hmm. And then until, of course, this new next generation sequencing came along, then I instantly realized that this is the solution, perhaps. So, and we published, I think in 2010, we published a paper with my associate on the use of next generation sequencing as a replacement for MAPRIC. And it was just remarkable. I mean, you can quantify the mutation at 472C in type 3 OPV and quantify it exactly, get exactly the same numbers as with MAPRIC. But also you could identify the entire, as we call it now, SNP profile. So you can measure the content of every type of mutant in every single nucleotide in the genome. So it generated enormous amount of information. So we immediately realized this could be a solution, and we proposed in that now 13-year-old paper, we proposed that it could be a replacement. And then we started collaborative studies, again, under the WHO auspices. Mm-hmm. And it was accepted just last year, or a couple of years ago, it was accepted as a replacement for MAPRIC, because now it's much easier to do. Basically, it's a very well standardized method. Companies, they don't want to mess with all this equipment and know how they can contract it out. Right. I mean, there are a lot of companies that will do it for a living, basically. They have highly proficient and so on. So, but now we're still working on replacing the monkey test. Right. Because and we presented this concept last year at the Expert Committee on Biological Standardization at WHO, suggesting that looking at this SNP profile tells you much more than the monkey test, because monkey test is not very sensitive, and it looks on just one phenotype, but there may be some other things. For instance, mutations can disrupt antigenic sites, mm-hmm. and it's also a matter of consistency, right? So, and the ECBS, they were quite positive, and they say, it may be a good idea to just go ahead and organize a collaborative study, get everybody to learn how to do it and harmonize and so on. So now we're in the process of doing this, and hopefully this will be, I think the plan is currently like next year to wrap up the collaborative study, mm-hmm. And come up with the criteria for pass-fail decisions, because it's not trivial. And it's a heavily bioinformatic task. Essentially, you have roughly 7,500 nucleotides, and each nucleotide can have 3 different types of mutations. So we're talking about 21,000 mm-hmm. measurements. How do you make your decision based on 21,000 analytes, quantitative results? So,
well, you can use your viruses that passed or failed the monkey test to generate some baseline for that, right?
Not really, because every company makes their vaccine with different profiles. Okay. Even though theoretically they come like 2 passages from the master seed virus that is stored at the WHO. still, conditions, even one passage can change significantly. So that's why one solution would be for each company to establish their own baseline of this profile and then stick to it. But the question is how much deviation you can afford is acceptable. What if a mutation comes up? How to decide whether it's significant or insignificant. When you have to switch to a new seed virus, which happens because you run out of the old one, you make or you change conditions. Something you import you install a new bioreactor with slightly different conditions. How do you make sure that this is still acceptable? So there are a lot of questions that may look trivial for those who are not involved in this regulatory field, but somebody must have an exact SOP how to do it, and the SOP for not only technical, but how do you make your judgment about the acceptable, unacceptable. So and it's quite, that's what they call regulatory science, answering all these questions that are, for most people are mundane and uninteresting, but without answering these questions, you cannot make biological products.
Sure. All right, let's end up with eradication. So 1988, WHO said we're going to eradicate polio. And now today we have no more disease called by wild types 2 and 3. Only wild type 1 continues to circulate. But we have replaced the types 2 and 3 with vaccine-derived viruses. So, and in fact, there's more polio now than we had previously for many years. So, and it's all caused by vaccine-derived virus. So, what is your outlook on eradication? Are we able to eradicate? And there are two questions. I mean, one is, can we eradicate poliomyelitis? And the other is, can we eradicate poliovirus? And what was the original goal? Was it poliovirus or the disease to be eradicated?
Well, my favorite response to this is to say Bill Clinton. It depends on what your definition of the word is. Indeed, I mean, what does it mean to eradicate virus? Actually, there is a very clear definition that if you use vaccine and you control disease at a certain level that is everybody thinks is acceptable, that's a control. Then elimination means that you have no cases.
Correct.
But you only can maintain this zero morbidity if you continue to apply measures of hygiene, vaccination, and so on. So this is a situation when you basically have no disease, but you must be vigilant and do everything to maintain the status. Eradication means entirely different thing. It means that you eradicate, you uproot it completely. Basically, there's no way virus can get back into circulation, so you can stop taking protective measures, you can stop vaccination because there's no virus and it will never come back.
That's smallpox situation,
That's smallpox,
right?
even though in retrospect it's not so, because there was a good reason why at the turn of this millennium they had to develop a new vaccine and put it in a stockpile in case it comes back, because it's still there. Not only it's there, it can be easily synthesized now.
Right.
So that just for this reason alone, I think that in the strict sense of the word, eradication of polio is not possible. But I'm not going into semantics, and I'm not going to argue. Let's raise this issue and say that it's not possible. I think it is possible in a common sense of the word. That eradication means there's no disease. And actually, when the WHO, World Health Assembly, resolved 35 years ago, they resolved to eradicate polio, they meant exactly that. Because if you go back and read this declaration that was, I think it was signed on May 13, 1988, 35 years ago, it says that we resolve to stop poliomyelitis in the context of the improvement of vaccine delivery of all other vaccines. So the idea was that we can eradicate poliomyelitis. There was not a single mention of eradicating the virus, not even once. by improving vaccine coverage to 80%, and they even call the exact number 80%. I think maybe now we should adjust it to higher than 80%. But at the time, their belief was that 80% should be enough so that there will be no polio. But very quickly in the program that was established, this Global Polio Eradication Initiative, there was a mission creep. They basically interpreted it as a hunt for viruses.
I see.
And they set the unattainable milestone for themselves. Basically, their measure of success was no virus, no circulation, nothing. Basically chase it down to the last virus particle. And we know this is extremely difficult because the disease itself is sort of, benign. It just paralyzes 1 out of few hundred or maybe 1,000 people. For those who are paralyzed, it's not benign at all. It's a sometimes deadly disease, a nasty disease, and leaving people paralyzed for life. But in the public health sense of the word, it's not really a big problem. It never was until the turn of the 20th century when it became pandemic. With the vaccination programs, it's also not a huge problem in countries that can deliver vaccine, but in countries that most kids are not immunized, of course it's a problem. And this is a problem for public health authorities. Why don't we immunize everybody? Why don't we improve vaccine delivery and instead chase this ghost of this eradication of one disease that maybe a few hundred cases a year? In some years it's even less. This year it's even less than close to, we're not talking about 1,000 cases in the entire world. So, but still the objective of the program is unchanged from 35 years ago. And the definition of Albert Einstein. What's insanity? It's doing the same thing and expecting a different outcome. I'm not, saying that people who are doing this are insane, but collectively we are insane. Trying to do this over and over again. So, we published a lot of editorial papers with Eckhart Wimmer, Eli Ehrenfeld, Vadim Agol, recently with Bob Gallo and Stanley Plotkin and Christian Brichot, arguing that we need to go back to the drawing board and rethink, what we're trying to accomplish. No response. No response. It's just... So that's why one other reason why you say, should I continue fighting windmills? That's... Well, let's see. I wish it happens, but I'm not optimistic because a virus can hide in a lot of places. There is also this phenomenon of orphan lineages, meaning that in an area where polio was kind of declared eradicated, and 5 years later, virus is isolated with genetically traceable to the lineage that was circulating here 5 years ago. So, when you bring this argument, those who are kind of arguing for this continuation of this, say, oh no, it's just a failure it's a kind of surveillance breach. So what does it matter? Surveillance breach, it can hide from us, it can circulate undetected for 5 years. And you say that after the last case of polio in 3 years we can stop immunization? Does it make sense? How you can make sure that there is no cryptic circulation? Okay, you can look in the wastewater in places that have this sewage system. And that's how this recent event unfolded in New York City, London, and Israel. But there are plenty of places on this earth where there is no sewage system. So how can you make sure there is no circulation? And that's exactly where the virus is circulating. It's impossible to do environmental surveillance worldwide at quality that would be sufficient to make sure that there's not a single virus particle remaining in circulation. So that's why I think that the solution is very simple. Just improve immunization, vaccine delivery everywhere. And fold this IPV, or perhaps use this novel OPV that was developed into the routine immunization schedule, depending on the local circumstances. Because if everybody is immune, who cares if the virus is circulating or not? And if there is one case, okay, well, it's very unfortunate, but it's real life. I mean, you cannot really throw billions of dollars just to chase something that you can never accomplish. I don't know. I mean, I wish them success. I wish that happens one day or they declare it. But again, I don't know what it means. I don't know if we want to do this. Do we really want to create a world where everybody is a hostage? Can you ever stop immunization? I think no. Never, ever. I mean, it's just unacceptable, because if you stop immunization, in 5 years' time, every child under the age of 5 will be susceptible. Sure. And can you imagine the tragedy that will happen if some insane person would synthesize it from chemicals, which can be easily done in a week, right? I mean, can you rule it out? No. Mm-hmm. So should we make every child a hostage to the situation? Of course, it will be just ethically unacceptable. So that's why I think that immunization should continue indefinitely. And when the current vaccines are not perfect. I mean, even recently, sadly we learned even this novel OPV that is by far more stable genetically than the original one is also not perfect. It also can mutate at a much lower rate, but there are so many new ideas, new technologies that can result in a much better vaccine. Mm-hmm. So I think it would be unacceptable not to do it. I mean, I know that maybe economics is not there, because with this number of cases, it's probably nobody would want to develop a new vaccine. But if you think strategically, I think it would be much better if we had a very inexpensive vaccine that would be induce comprehensive immunity. But unfortunately, I mean, this is not a perfect world, and we probably will have to settle for something. But even the existing IPV, if it's used in the combination with other antigens, such as DTP, this pentavalent, hexavalent vaccine, which now some companies are developing, and already these vaccines are being used. If we put more efforts into developing a worldwide infrastructure for vaccine delivery, that would go much longer. First of all, it will promote immunization against other diseases. So it will be kind of win-win. It will be a kind of effect that would be not only on this polio, which doesn't really have a big economic impact right now. I mean, we're talking about a few hundred cases a year. So that's why I personally think that this eradication initiative is completely misguided. I mean, it was misinterpreted, the original intent. And now I think that it's a good time to go back to the drawing board. But who am I to advise it?
I think you've thought about it a long time. You are the right person. But I think your idea of developing new vaccines makes a lot of sense. But we're being told to stop working on polio. So can it be done in a containment world?
Of course not. Of course not. But the sad thing is that when an initiative gets into the hands of bureaucrats, that's the end of the story. Because, I mean, at WHO, the left hand doesn't know what the right hand is doing. I mean, one part, which I'm participating, I'm a member of the Polio Research Committee at WHO. This is a group of people, they understand all these problems. And they do all their best to help development. This committee is reviewing grant applications for some projects and vaccine development, surveillance, and everything. So this is a group that is trying to do the right thing. But there is another group that is actually doing this containment. They're deaf to all the arguments. I mean, because they're trying to do their job. Their job is to stop polio research. That's it.
Right.
They want to stop it because they were told that it's not needed anymore. Somebody told them way back, and now they're on a mission to stop it. And then it is WHO is not maybe the biggest problem, because now it's delegated to each country, and at CDC the same thing. They don't care if that's an important project. They're just on a mission to stop it. So that's why I'm not optimistic that it will ever happen. And my pessimistic scenario is that they will declare eradication and then something really bad happens 10 years from now. I mean, So we will remember the COVID outbreak as a kind of cakewalk.
Yeah, if you stop vaccinating, as you said, that'll be a real problem.
It's not. It not can be. It will be a problem.
So you, despite retiring, you're still involved with these questions, right? You said you're part of this WHO group.
Right. No, I'm still involved because I strongly feel that this development of this next generation sequencing approach, it's not only important for polio because it's a kind of new paradigm for quality control of all vaccine products. Because for most vaccines, you don't have such a costly animal testing on So most vaccines are not tested. But the issue of consistency is very important. And not only for vaccine products, but also for other biotechnology products, such as for gene therapy and so on. So I know that for some experimental product development, they use something similar. So I think that getting it through the entire regulatory approval pipeline is very important. It will be a new paradigm for quality control. That's why I'm still involved, and I want to help as much as I can, even though I don't have a wet lab anymore, but I have some expertise in both in bioinformatics and virology. So I think that is important for developing of this new approach and interpreting results and kind of, 33 years being at the FDA, I learned a few things about regulation.
Could one make a polio vaccine, is not injected, it is not infectious, but which you would take orally and would immunize your intestines and generate immunity?
Yeah, I think it is possible. It is possible. There are different ways to do this. Before my retirement, we started, for instance, working on the RNA vaccine against polio and got very good results in vitro. We didn't finish it, and hopefully somebody can continue it. But we had excellent, after transfection of cells, there was more antigen, the antigen in cell culture than after virus infection. So it clearly can be done.
Yeah.
So you can if you can deliver this RNA, then it will be a good vaccine. Of course, there's a long way to go.
Sure.
Yeah. Another, I think, approach that I like very much is that George Beloff proposed that he expressed polio capsids in NDV, Newcastle disease virus. It can be other virus, but also with very good results. Good VLPs with the antigenicity are expressed and can be delivered on mucosal surfaces. So in theory, you can make a vaccine that contains no live virus, and that can most likely can produce mucosal immunity, similar to oral polio vaccine. So there are many ways to do it. Also, I work on adjuvants for inactivated vaccine.
Yeah.
At some point, we worked on vitamin D3 that is known to redirect dendritic cells into peripheral lymph nodes. We had good results in mice. Never tested in humans, just because somebody else pursued it for flu vaccine, and despite good mouse results, in humans they did nothing. Perhaps a difference in the vitamin D3 metabolism. There are many ideas. But without being able to work with polio, nothing would come out of it.
So I have one more question for you. If you had not been a scientist, what would you have done?
I don't know. It's very hard to. Look. And I'll tell you that I grew up in the Soviet Union. As the way I looked at it, it was the only way to live a kind of relatively decent life and not make moral and ethical sacrifices. Because what else could you do in the Soviet Union? What was the other career? To become a party functionary or become a military guy? Or there wasn't. When I was a little kid, I dreamed of becoming an airline pilot. But boys dream about stupid stuff. And maybe it's not such stupid, but I was fortunate that I grew in a family that actually kind of subtly channeled my intellectual energy into something.
All right.
All
right, that's a special episode of TWiV. You can find the show notes at microbe.tv/TWiV. You can send questions or comments to twiv@microbe.tv. If you enjoy our work of spreading the word about science to everyone, please consider supporting us. We exist on your financial support. You can go to microbe.tv/contribute. My guest today, Kostya Chumakov. Thank you so much. Fascinating career, great stories, and I think the polio story is not over yet.
Yeah, I hope so. I hope so.
Thank you very much for joining me today.
Thank you, Vincent, for having me.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.